Good afternoon, everyone. Thank you for coming. By way of introduction, my name is Jess Thompson. I'm the chair of the CRF Research Committee and work with the Scientific Advisory Board. It's my pleasure to be able to have our researchers here today. And our first, sort of the thought that we would have is we would give the talks, and then if we have time, there might be time for one or two brief questions and answers. But otherwise, probably what we'll do is after we give the talks, we'll bring everyone up and kind of have a panel sort of discussion. So if you don't get a question in right after the talk, that'll be okay, because if we have time at the end, we'll bring everybody up and we can sort of all ask questions. We're going to have, most of them are going to be live talks. There's going to be a couple of talks that are pre-recorded. And so I'm very excited for what we're going to hear this afternoon. Our first talk actually is going to be by Ben Shaberman. He's the Vice President of Science Communication for the Foundation of Fighting Blindness. And he's been in this space for over 20 years. And he's going to talk about the latest news on emerging retinal diseases and therapies and local and national events for patients, families, and eye care professionals. He also conducts a variety of scientific education activities for staff and constituents. And with that, I will turn the microphone to Mr. Shaberman.
Thank you, Dr. Thompson. Can everybody hear me okay in back? Excellent. Thank you. Well, it truly is an honor and a privilege to be here at the CRF meeting. Dr. Thompson and I were talking about when I began more than 20 years ago, there were very few clinical trials, virtually none, for any retinal diseases. And CRF was a small, humble group. And now we have more than 50 clinical trials underway in the retinal disease space. And I am so impressed at how well CRF has grown. You have become a real force in driving research and bringing your community together to try to get treatments and cures for choroideremia across the finish line. So thanks for all you're doing, and congratulations on your growth and success. Clearly, we still have more work to do. A little background on the Foundation Fighting Blindness, if you're not familiar with us. We've been around for about 50 years. We've raised more than $1 billion toward research for inherited retinal diseases like choroideremia and retinitis pigmentosa, Stargardt disease, and others. We currently fund about 100 projects in the US and overseas. And today I'm going to cover what I think is kind of an eclectic mix of projects that are related to choroideremia. And as you heard, we'll have an opportunity for questions after others present. So I think many of you are aware of the Biogen gene therapy that came from Nightstar, that originally came from the University of Oxford. This gene therapy for choroideremia did in fact have an effect, a positive effect on vision for the people in the trial. Unfortunately, it didn't meet its primary endpoint of visual acuity, or best corrected visual acuity. And that's been an ongoing challenge in the retinal disease space. So we are actually funding a young clinical researcher out of Rob McLaren's lab. Rob is obviously at the University of Oxford and a founder of Nightstar. Her name is Dr. Maram Abdallah. And instead of using visual acuity as an endpoint, she's looking at a structural endpoint, looking at the actual retina itself. And it's an area in the macular region called the smooth zone. And this shows where there are remaining photoreceptors and where photoreceptors have been lost. And you look at the smooth zone, as I have on the slide, through a fundus image, through autofluorescence. And autofluorescence fluoresces the areas where there's some degeneration and it also demarcates where photoreceptors and vision have been lost. Anyway, based on some initial analysis, Dr. Abdallah and the lab believe this is a more sensitive and precise measure of efficacy. And so if there is a future gene therapy clinical trial for choroideremia, this may be a better outcome measure. And I did read, in looking at her application, that Oxford actually has gotten a license back to that choroideremia gene therapy. I don't know what their plans are in terms of getting that into a trial, but a good endpoint would definitely inspire investment in getting a clinical trial off the ground. And we are giving Dr. Abdallah a $63,000 grant to study this smooth zone. So I want to switch gears now and talk about an emerging approach to mitigate oxidative stress that may at some point apply to choroideremia. So what many different researchers who study retinal disease have observed in people with retinal conditions like retinitis pigmentosa or choroideremia, rod loss happens first. And the loss of rods ramps up oxidative stress. And what Dr. Campachero at Hopkins has concluded is that oxidative stress exacerbates the condition, especially loss of cones. And cones are what give us our central vision, color vision, vision in lighted settings. So if you can protect cones, that could be very powerful. So Dr. Campachero and his lab evaluated a very powerful antioxidant called N-acetylcysteine, which I will call NAC from here out. This is a drug that has been around for decades. It's FDA approved. It's used in Tylenol overdose. But he studied this drug in animal models of retinitis pigmentosa and showed by mitigating oxidative stress, you could really slow disease progression significantly. So after that work, with funding from the National Eye Institute, he is launching a phase 3 trial for NAC in dozens of sites in the US and also overseas. They're going to enroll more than 400 patients, and they're pretty close to finishing up enrollment. And they're hoping over a 2-year period to be able to tell if dosing with NAC in these RP patients will in fact slow vision loss. And in thinking about this trial, I contacted Dr. Campachero and said, if you get good results for people with RP, do you think this molecule could be relevant to people with choroideremia? And he said, certainly it would warrant further study, because again, the rod loss that leads to oxidative stress is also present in people with choroideremia. So we're hoping if this trial is successful in RP, we can leverage the results for choroideremia and perhaps other retinal conditions. Related to that, we are invested in we, meaning the Foundation Fighting Blindness, we're invested in a company called Nacuity that's taken NAC, modified it to be more potent and bioavailable for the retina. So the idea is at a lower dose, we're hoping you can get an even better effect. And Nacuity, that's the name of the company, they've been conducting a clinical trial in Australia now. Pre-pandemic, it began for people with Usher syndrome, which is essentially RP with hearing loss. And they should be reporting results very soon for this molecule, which they call NACA. And if those results are positive, they'll move to the US and launch some sites here for RP and Usher syndrome. But again, It's the same approach. It's just a more powerful version of NAC. So we're hopeful that that study will yield good results as well. And just to make it clear, because it's an antioxidant, it is a gene agnostic approach. It's not addressing a specific gene or mutation. So that's NAC and NACA. So now I'm going to switch gears a little bit and talk about gene therapy. And I mentioned the Biogen gene therapy for choroideremia. There have been dozens of gene augmentation or gene replacement therapies in clinical trials now over the last several years. And most of these use a virus, an engineered virus, to deliver the genes to the retina. And the most commonly used virus is what we call an adeno-associated virus, or AAV. And many of those trials, the therapy is injected subretinally. So what's literally happening is it's a surgery, and the needle is being put through the retina, and a little drop of liquid called a bleb is being injected underneath the retina. It lifts the retina up. It actually detaches it. And that's what researchers up till now have believed we've really needed to get a gene therapy to the cells that need it. The problem is it's invasive. It requires surgery. Retinas are fragile, even if they're not diseased. If they are diseased, they're even more fragile. And these subretinal injections are by no means trivial. The other approach to delivering gene therapy is through the middle of the eye, intravitreal injections. And while those are a lot easier because you don't have to inject into the retina, they can be inflammatory, and the therapy is injected further away or farther away from the cells that need it. So there are challenges with intravitreal injection. So that's sort of the background on what's inspired a researcher at Ohio State University, Tom Mendel, to look at a new and improved approach to gene therapy. And the approach involves taking out the vitreous, that gel in the middle of the eye. It involves pulling away a vestige of eye development called the inner limiting membrane, which can get in the way of therapies getting to the back of the eye. And then it also includes insulin to help the therapy get absorbed into the cells that need it. So it's kind of a three-step approach, and it's going into the middle of the eye, not subretinally. So he thinks this might be a much better way to get therapy to the cells that need it without having to go subretinally. And the last step in this process is after a half hour after the patient receives the gene therapy, if there's leftover vector, which can be inflammatory, they take that out. They suck it up, vacuum it out, however they get the remaining gene therapy out. So Dr. Mendel is studying this approach in mice and in pigs, and if it works, it's something that could be applied to a lot of different future gene therapies, including choroideremia gene therapy. And so finally, I just wanted to mention another project that we're funding. This is Dr. Bana Telugu at the University of Missouri, and actually he was supposed to be here. This was going to be my introductory slide for him. But since he's not here, I'll tell you a little more about this approach. This is the creation of a model of choroideremia in a mini pig. And we like these quote unquote larger animal models because their eyes are more similar to the human eye. So you can get a little more idea of how your gene delivery or other therapy delivery needs to happen. So we're giving him a $500,000 grant to develop mini pigs that have choroideremia. We can use these to not only study disease and natural history, but also as a way to test therapies like gene therapies. And they like using mini pigs because, like regular pigs, are obviously massive animals and much harder to maintain, more expensive to maintain. So stay tuned as that work moves forward. So I've given you an eclectic mix of projects here, but we're excited about the work that's going on for choroideremia. And thanks again for all you're doing to support the mission.